Okay, welcome to the clustering lab. So just to get started here, these are the packages you're going to need. Let's see, we have a few installations from Bioconductor, so you can run this code just as I've done it here to get all of these different packages. These two are from Bioconductor. These two are from CRAN. And I'm going to just assume that you're going to pause this and install them, and then I will mark these out, and I'm just going to load the packages. Okay, so the first thing we're going to do is we're going to do some basic clustering using k-means and k-meanoids. We're going to look at the selection of clusters using a variety of different methods. So let's just, first of all, we're going to take a look at the iris data. And this data is represented in many different packages. That's also a default here in R. So it's internal to that program, and basically if you do head iris, you will see this is like the data that we talked about in class. And if you look at the variables, there's only 4 variables, and if we look at the classes, we can see that there are 3 different classes of flowers, just like we discussed. And if you might recall, the Setosa was quite different than the other two, which have a lot of similarity. So hopefully we're going to see that in our clustering today. So let's go ahead and get started. Let me just put in a few of these commands. Iris and head iris. Okay. So let's start actually by applying k-means. Pretty simple. And so what I'm going to do here is I'm actually going to create, but maybe I should do it up here. I'm going to create a data set. It's going to be the iris data, except I'm going to only consider the first 4 columns. So that's my way of doing that. And now if I look at the first 4 columns, I see that my species column has disappeared as I want it to because I don't want to use that label in clustering. Okay, so let's apply k-means. So the first thing let's do is just take a look at what we have here for k-means. You put your data matrix in however many centers you want to have. In other words, what k should be. You can control the number of iterations. I typically don't do this. We can also use multiple starts. Okay, and there's some specific algorithms you can set. If you don't set anything, it will take the first one, which is just fine. It's standard. So let's see here. Let's call this km. I'm going to output the object of k-means. And I'm going to do k-means of the DATs that I just created up here. And I'm going to make it with 3 centers. In other words, k is equal to 3. And I want to do 10 starts, okay? Because I know that convergence is not guaranteed for this method. So let's just take a look. Whoops. Of course, that should be 3. Okay, so let's just look at the output, km. Okay, so we have a lot of information here. We have the cluster means. Okay, so there's 3 clusters, 1, 2, and 3, and their coordinates, their means for each of these variables are specified. We also have the clustering vector, which will tell us which observations are being set into which clusters. This is very similar to what we saw in class in the sense that we know that the Setosas are all at the top of the matrix there. These are all coming up in the same class, and then we start to see a mixture of the other 2 varieties of flowers. We have some measures here about within a cluster sum of squares, and that's giving an impression of the dissimilarity. And we also have fields that we can pull. So if we want to, for example, look at km clusters, we can just grab the cluster labels and so forth. The size of the clusters, how many iterations were processed. So we have all this information. So let's take a look at how well does the clustering match the labels? Okay, so of course we're playing the game here that we don't know what the labels are when we do the clustering, but then once we get the clusters, we kind of want to take a look back to see how they represent the original set of labels. So first thing I'm going to do is I'm just going to tabulate KM cluster. Okay, so that's going to pull out my cluster object. And now I want to look at iris and species. Okay, so let's just take a look here. Okay, so we can see here actually quite nicely that the Setosa all clusters together in cluster 1. This is like the NCI slide that I showed you with the different cancers falling into the different clusters. So we see very good purity here. In cluster 2, we see a mixture. It's largely represented by this versicolor species. And for Virginica, we also see a mixture, but this is primarily Virginica. So we see some, this one, these 14, and these 2, these are kind of representing those pieces that are a little bit in the middle, okay, between the two boundaries here. So let's actually visualize this. I'm going to call it the groups, okay? So I'm going to use Quartz today. I'm on a Mac. You could use X11. So I want to plot actually the data. And I'm actually going to plot just sepal length. I could use the column indices if I want to here, but I can also use the variable names in this case. And I also want to look at sepal width. And that'll come into light as to why I'm doing that, because I have, of course, 4 variables that I'm clustering over, so I can't have this 4-dimensional plot, right? So actually it turns out that sepal length and sepal width are the most important variables in discerning the groups, and we're going to see that in a few minutes when we get to the PCA. So let's do this. Actually, we need to do it like this. I missed a bracket here. Okay, so I'm including all the rows, all of the samples, and I've got my sepal length and sepal width. Okay, and what I'm going to do is I'm going to color it by cluster. So in order to do that, I can set the color KM cluster. Okay, this is coloring it by the assigned cluster. And for my title, which I'm going to call main, this is going to be called example k-means. Okay. And the other thing I want to do is I want to actually plot the centers. Okay, so not only do I want to color it by group, but I might want to also plot the centers. So let's see here. It doesn't seem like I have one too many brackets. Okay, so I'm going to first let's plot this and just take a look at what it looks like. Okay, so this is what it looks like. We have sepal width. See if I can shrink this. Sepal length and sepal width, and we can see the 3 groups, but I'd like to understand where their means are calculated. So I want to actually plot the mean here somewhere. So I'm going to add a layer to this plot, and to do that I'm going to use points, okay? And I'm going to plot the centers. So KM centers, okay? But of course, I only want to include sepal length and sepal width, right? So maybe I can just copy and paste these 2. Okay, I don't want to include the other measures, petal length and petal width, because I'm not using them in the plot. So here we go. All right, and now the color, I'm just going to use the first 3 colors because that's what's being used here. And PCH is going to set the size, so I want it to be a little bit bigger. Okay, so let's go ahead and plot this. Okay, so this looks pretty good. Now we can see where the centers are. So you can see that they really are centers. And we see these mixture points. These are the ones that are sitting on this off diagonal here. Okay. All right, so let's do a little bit more. Let's talk about the RAND index and the adjusted RAND index. Oops. Calculate RAND index and adjusted RAND index. Okay, so there's lots of ways to do that. I'm going to use the functions RAND index and this is from the fossil package, which was also featured on the blog that I shared with you. And the adjusted RAND index can be calculated in this package as well. You just need the two groups, okay? So these are all the details that are there. So in this case, we have our two groups that we want to compare. One of them is going to be the KM, right? Oops. KM cluster. And the other one, so for this one, we want to use the iris species. However, the issue is this is going to give us a vector of factors. And, you know, we can't really compute with that. So if we actually do as numeric iris species, then we can convert this into a numerical string for which we can contrast it against this. Now, because of the way this is computed, it doesn't matter. These are, you know, different strings and so forth. There doesn't have to be a coordination between how these indices are set. It's really what is common between the indices within each group. So in this case, we don't have to think too much about it. We can just use as numeric iris species. Okay. And we're going to do the adjusted RAND index just this way as well. And the only difference is I have a command here that's a little different. So let's take a look at this. Okay, so we have the RAND index, which, you know, is pretty high in this case. And the adjusted RAND index is a little bit more conservative, but they both look pretty good. And this is for having 3 centers. And we know that this is the true number of groups, okay? So we could also, you know, practice taking this down to 2 and all kinds of things, but I kind of want to move forward from here. Okay, so let's take a look now at maybe switching things up just a little bit. And we kind of see, you know, the fact that the sepal length and sepal width is going to be pretty important, and I want to show you how you can kind of see that. One thing that we can do is we can look, let me just save this so I don't lose anything. We can look actually at applying k-means to PC1 and PC2. Now I'm selecting these two because I want to visualize it, but what you're going to find out is that actually PC1 carries most of the information and PC2 is really not contributing too much. So the first thing we need to do is we need to come up with a principal component representation. So in order to do that, we're going to use fit as our object that we're naming, and we're going to use prcomp, which is the more stable way to do principal components in R, and we're going to apply it to our data without that column of species, and I'm going to center this, and I'm going to scale it. Okay. Because remember I said that the scaling is going to be important in terms of computing the principal components in this setting. So we always want to have these to be true. Sometimes we don't need to do the scaling, but we have to kind of look to see. Okay, so if I look at fit, if I output fit, I get information here about the principal components. But if I look at names fit. This is everything I have. And so the loadings are stored in here in the rotation. So I can access the loadings by fit rotation. Okay, so this is the loadings. And what we can see here automatically is that the sepal length and sepal width seem to be pretty important in terms of these are the ones that are further apart, divided by, straddling 0. So these are the ones that are related to the most variation in the data set because they are kind of the furthest apart, talking about the spread there. These are also having high magnitude in this setting, but they are close to each other. So there's really nothing going on here with the first component in terms of dividing the petal length and petal width apart. So it has a less contribution to variation. So let's just plot. If I look at the variances here, this is what I have. Okay. Now, incidentally, I won't show you this, but if you decide to take scale off, make that false, then you might have something that looks really weird. Like this becomes really, really, really big and everything else is small. So you have kind of a scaling problem when you see something like that. So try that on your own. You'll see it in practice, but we're going to move on here. So what I want to do, let's just repeat what I did here. Quartz fit, oops, sorry, plot fit. Then you're going to see how healthy your scree plot looks. Okay, let's just take one more look at it. So we can see that this is obviously the dominating factor. This is also important. But if we use that one criteria, we would probably drop this one as well because it's below the measure of unity here. But in practice, we're going to use these two because if I look at the scree plot, I see this is definitely the noise floor. So we're going to proceed with the first two principal components. We know that. But before we do that, I just want to take a look. I'm sorry, I don't want to use plot. I want to use biplot. I'm going to look at the biplot to have an impression of what the variables are doing here. And this is what you see. Okay, and this is, you already saw this from looking at the loadings, but we see these are our scores. These are all the 150 different measurements of the various species. We see this is probably the Setosa, and this is the mixture. And what we have is, this is the first principal component explaining the majority of the variation. So the majority of the variation is explained by sepal width and sepal length. Petal length and petal width are very close to each other, and they're very similar. So there's not much distinction here. And they don't really do a good job at separating these various classes. So for discrimination, they're not very useful. And we also see, if I look at the PC2 axis in terms of what's dividing this plane at 0, I see that I really don't have much here. Okay, so PC1 is obviously the most important, but we're going to cluster with the two principal components. So PC1 is going to be fit X, and it's going to be, oops, the first component. PC2 is fit X, the second component. So if I do, again, names fit, this is the rotation, these are our loadings, but now this one is big, so I'm just going to show you the dimensions. Dim of fit X, okay? So these are the scores, okay? These are the scores of the various items on the principal components, and these are what we're going to be using instead of the variables. So obviously there are as many columns as there are variables in our original data set, and here are the 150 samples. So I'm using the first principal components in place of the original data. This is PC1 and PC2. Okay. So now I'm going to create an object. I'm going to call it PCDATS. And this is going to simply be cbind PC1 and PC2. Okay. All right. Oops, I forgot to run this. Okay, so we have that. And now what I'm going to do is actually run k-means on the PC values. So in this case, we're just going to use the same thing we did before. I'm going to call this KM2. It's a new object. My other one was KM. And it's going to be k-means, and it's going to be on PC DATS. And I want it to have 3 centers. And end start is going to be 10. Oops. Okay. So that's my KM2. And what I want to do here is I'm just going to copy and paste this. Okay. I'm not going to copy and paste the header, but I'm just going to calculate the RAND index for KM. Actually, this is going to be somewhat redundant, but KM2, KM2, okay, because that's going to have my new labels based on the principal component. So let me just get this in my workspace so I can remember what the RAND index was in this case. Now I'm going to run k-means on the principal component data, and I'm going to look at the RAND index and adjusted RAND index so I can compare these things. I can see actually that using the full data is going to give me a little bit better performance in terms of agreement with the original data. This isn't altogether too surprising. So why would we do principal components on a data set of this type? Well, you probably wouldn't, because you don't have a situation where you need dimension reduction or you need to decorrelate the variables, but still we want to go through the exercise because it turns out that those other variables don't actually contribute much. So it leads to a more parsimonious model, and we don't see a whole lot of difference here between these two values. And again, we're talking about a situation where we want concordance with the labels, but this is not a classification problem, so performance isn't necessarily something that we're striving for in terms of getting these values correct. So the next thing I want to do is I want to plot the groups, and I think what I'm going to do here, let's see, plot the groups. So we start off in the same way, and we're going to plot PCA DATS. I keep wanting to say PCA DATS, PC DATS. And we're going to use the colors from KM2, our second clustering cluster. And we're going to have example k-means with PC. Okay. And we're going to do the same thing here where we have our points. Our points are going to be coming from the centers, KM2 centers, okay? And the color is going to be 1, 2, 3. And I'm going to set the PCH to 8 and the sex, which is controlling the size of the font, to 2. Okay. So here's our solution, and you can see it actually looks a little bit cleaner. So although we did not see the adjusted RAND index, clean up as nicely, have as much concordance, what we see visually is a better separation here between these groups. And if we look, for example, at a silhouette plot, which we will shortly with k-medoids, we might also see that this is a better representation. Okay. All right, so let's move forward. Actually, let's move on to k-medoids. Okay. So the k-medoids routine is going to be executed with a function called PAMK. And this is the information. It's in the FPC package. And I'm just going to show you the basics here. So k-med, and it's going to be PAMK DATS. So this package is not necessarily well documented, But once we apply k-medoids, there is a function, so I'm going to say let the program decide optimal k. Now in the help file, they don't exactly tell you what they're doing here, which is why I only want to spend a moment here on this. Okay, so PAM-K tells us that there are two clusters, whatever optimal criteria they are using here. They say that there are two clusters in this data set. We're going back now to the original IRIS data. Okay, so let us now tabulate the results just as we did before. So we'll do a table, and this is going to be k-med. And the way we're going to get this, you could do names k-med to see what is involved here. There's a lot of different outputs, but if you want to get to the clustering results, you need two levels of output. And IRIS species. Okay, so we want to just tally these things up. Okay, and so here we see that if there are two clusters, which k-medoid says that there is, what we have, if we tabulate the results, is we have Setosa in one and these two in the other, which we would also expect based on what we know already about this data set. Okay, so now let's plot the results for k is equal to 2. And actually, there's a really nice plot associated with this, this function. So k is equal to 2. Okay. So here, what I'm going to do first of all is create a layout for my plot. And the layout is going to be a panel of two plots. So that to do this, we can use layout matrix. And within this matrix, we have C1, 2. We have a 1 and a 2. Okay, so this is what is going to give us the proper layout for this particular plot. Plotting is a little bit of an art here, but then the rest of it is easy. We want to plot k-med, the PAM object. Okay, so let us take a look here. Beautiful. Okay, so this is our plot, and you need to kind of reformulate it here. So what it's doing is it's plotting actually on a principal component plane for visualization. And these are the two clusters in the principal component plane, which is similar to what we saw already. And then what we have here is a silhouette plot. And so we have here average silhouette information. We have the number of points in each cluster and their silhouette value. And if we wanted to, we could also look into each of these individual ones. So that is for k is equal to 2. And let us actually try k is equal to 3, okay, and see what happens. So let's try k is equal to 3, okay. So the first thing we have to do is we have to run, oops, that's the wrong number. We have to run the method again. So k-med, I'm going to call it k-med 3. This is going to be the PAM-K for our data, and we're going to have 3 groups, okay? And I want to do the same thing I did before, where I'm going to table them. So I'm going to copy and paste this. I'm going to make this k-med 3. Okay, so this is what we have. It looks pretty similar to k-means. Okay, remember this is the off diagonal. That's where the mistakes were being accumulating. So now we have this information, and you know, we might want to also plot this. So let's plot this. Okay, so first of all, I need to refresh myself to see what the silhouettes were at for k is equal to 2. Okay, So this is for k is equal to 2. The silhouette was about 0.69. That was the optimal value selected by the k-medoids algorithm when we did not specify the number of clusters. If I specify the number of clusters at 3, let us see what things look like here. Okay, now I have something that looks like this. These are my 3 clusters. And we can see there's good separation here. The silhouette goes down all the way to 0.55, so not great. In particular, these 2 clusters do not look as good. So in this case, k is equal to 2. And if you forgot about the labels, you would probably say that that is a very good clustering. And it is. Okay. So now we're going to try a couple of other methods that kind of focus on cluster selection. So I'm going to create this break here. Let me save this. Ooh. Some other cluster selection that we talked about in class. Okay, so we have here, we did look at the silhouette a little bit with k-medoids. You can also look at a silhouette plot with k-means. We can look at the bootstrapping approach that I discussed. So that's going to be in the cluster boot, I think it's called cluster boot or boot cluster package that I loaded earlier. So there's a function there called kselect. Okay, and so kselect, there's 2 schemes here that I showed on the slide. One is taking the original clustering as the so-called true clustering, and we're trying to bootstrap the stability of that clustering. The second one is to search over different clusterings for an optimal clustering. So let's try both, and I'm just going to simply make it like this, kselect, and I'm going to go back to my original data here, just like I used with k-medoids. And I'm going to cluster over the range 2 to 6, and I'm going to do 50 bootstrap samplings with R, which is restarts, 5 restarts each. And I'm going to do scheme 2 is equal to true. So in the first one, I'm going to search for an over the bootstrap samples for an optimal clustering. And the second one, I'm going to let the original clustering be the gold standard. So let's take a look. Whoops. Scheme 2. Oh, I forgot my underscore here. I have to be careful with my syntax. Okay, let's try again. Oh. Now let's try again. Okay, so it's computing over these bootstrap samples, and let's see what comes out of it. Okay, so this one is actually saying that there are 3 clusters here, and that's using the original data, so that's good. This is actually getting the number of clusters correct in this case. Okay, both of them actually agree. So now let's try, so this one was actually bootstrapping, cluster stability. And the next one, let's try the gap statistic. Okay, so the gap statistic, it's going to be using a package called clus-gap. It's in the cluster package, and you loaded that at the beginning, and it gives you everything that you need to know here about the gap statistic. This is the stuff that we talked about in class that the formula is made out of. So it actually works on PCA rotations, which we did not discuss. So this is a better way to capture the variation of the data set when computing these different reference distributions. So the output is pretty comprehensive. Okay, let's see. We get all the information about these different values, and we're also going to be able to get a nice plot of the results. So let's just take a look. Okay, I'm going to call it gap k-means. So this is called clus-gap, that's the function, and we're going to use our DATs, we're going to use k-means We're going to look at the number of starts is 20. And it doesn't, you can put in any value here as long as it's greater than, 2. K dot max, how many do you want to go up to? We don't want to go past 10. That's a lot of little groups. And let's create 100 reference samples. Okay, so this is quite quick actually. Okay, now let's take a look at the results. So quartz, plot, plot, gap, k-means, okay, and my main is going to be gap statistic, k-means, okay. So let's take a look. I already ran that, so okay, this is our plot of the gap statistic, and remember we want to look for the max, and we also want to take into account the size of these standard errors and the shape of the curve. So here it actually looks like you would take your k to be 4 because it's large, it's larger than these certainly, but if I start to move out from 4, I start to see some increase and then some decrease, but I don't see anything that is one standard error or even slightly less than that difference from this one. So this one is actually saying that k should be 4. And now let's try the same thing. So the bootstrapping as it's set up right now, the one I just showed you, only works with k-means at this point. We do plan to extend that in the package, but we haven't done that yet. So let's try the same thing here, and let's look at, so this was gap statistics for k-means. Let's look at the same thing for k-medoid. So now instead of using this mean calculated from the data, it's going to be using a data point to represent the centroid. And so the only thing that we have to change here is going to be, sorry, this has to be PAM because it needs to know what function to call. So PAM is going to be the one that is used here. And this I want to change as well to k-medoids. And I might as well get this over with. We can do the plot right after. This is for k-medoids. Okay, so the other thing we need to do is, and I just found this bug, I had to pause the video there, but I have to eliminate the number of starts because PAMK does not have that as an option in its thing. So what I'm going to do here is I'm just going to rerun, I'm going to run this. Okay, it's going here. And this is the gap statistic for k-medoids. And, actually it's kind of saying the same thing. It's pretty much saying that k should be 4. You could say that k should be 3 in this case. I'm not sure if that's quite one standard error. If you were going by that rule, it might not. It may be overlapping indeed, so you might go with 3. But anyway, somewhere between 3 and 4. So, so both do a pretty good job, I think. The gap statistic, looking at a bootstrapping approach. There's lots of different options here. We could do silhouettes for these things. We could try to make some conclusions based on the adjusted RAND index. So there's lots of different things that we can do. The last thing I want to show you here is how to do hierarchical clustering, and I'm not going to spend a lot of time on that. Okay, so what I'm going to do here is I'm actually going to look at the GoLoop data, and in this case, let's see here. Actually, let us start with the iris data again. So in order to visualize this, and just because I want to be able to visualize it, I'm going to actually create a smaller subsample of this data. Not something I would typically do in an analysis, but what we're trying to do here is just kind of get a feel for the functions and the visualizations. So I'm going to sample the first column, okay, and I'm going to take a small sample of 60, okay? So this is actually taking a small sample from 1 to the length of the data in the first column, which will give you 150. I'm going to take a subsample of 60. So if I look at my IDX, this is my subsample, and I'm going to create an object called DATS reduced, DATS red, and it's going to be my original data with just the rows from my sampled indices. Okay, so there's my reduced data. And what I want to do is just calculate the dissimilarity, or I'm going to call it the distance here, for hierarchical clustering. Okay, so the function that we're going to use is called dist, and there's lots of different distances. I showed you that. So you do a help dist to see what's involved there, but I'm going to use the Euclidean distance, which should be the first one, but there's lots of different options we can choose here, and we can also write our own distance. So I've got this, and I want to just kind of look here, kind of wondering what the dimension is for this, because if you want to know, what am I clustering here? Whoops. Let's start this over. This is, sorry. This is DAT red, reduced data. Okay. Add an extra X. Okay, so this is 60 by 60, which means we're actually clustering here the sample observations, right? So you have to be careful because you want to make sure you're clustering the right dimension of your data. We don't want to cluster our 4 variables. We want to cluster the samples in this case, although there may be situations that you do want to cluster the variables. So doing a quick check to see what the dimensionality of the distance matrix is will tell you what you're clustering on. Okay, so now let's go ahead and do some clustering. So I'm going to call it HC for hierarchical clustering, and we're going to use hclust. And we're going to cluster using our dissimilarity matrix, and the method is we're going to use average clustering. Okay, so you can change the method and use some of the methods we talked about in class. And now what I want to do is I want to plot my result, HC. Hang is going to tell them which way I want this thing to actually hang in terms of up and down or left and right. And the labels I want to put on this, remember, maybe you want to put the species labels in this case. Maybe if you're doing another thing, you want to put the batch number and so forth. So we can use the labels and the same plot to kind of reveal some nuances in our data set. So let's take a look here. Okay. And this is what we have for our dendrogram. So we know that this is our Euclidean distance, and this is done using average linkage. And I'm looking at this, it's a very healthy looking dendrogram. And you can see that if you had 150 things here, this is why I subset it, only because I want to show you the visualization. But if I had so many things here, it would be difficult to start to read these things. So if I'm looking at this, it looks like a pretty healthy clustering. I see setosa in one group. This is what I expect. I also see some good concordance here in these 2 clusters. So if I'm looking at this, I might say that there's 3 clusters. And based on the shape of this dendrogram, I might take a cut over here at like 1.95 or something like this. Okay. So, the question is, how can you actually cut the tree? And so there's two things I want you to look at. Cut tree, and actually, even though there's two T's in that word, we have one T in the command, so, and then we have silhouette, which I always misspell. This is, the silhouette plot I just showed you before was built into the k-means or k-medoids package, PAM, but this is one that's standalone, and then we have a cut tree function here. So let's look at cut tree first. Okay. So we can do this in two ways. One is that, first of all, we give it the tree, which is our output to hclust. We can either specify the number of groups that we want to have, or you can specify the height using the picture of the dendrogram that you want to cut at. Okay, so, let's take a look here. Just one example. So let's say I want to calculate a silhouette plot. I'm going to call it SI. And I'm going to call this silhouette. For the silhouette plot, which I didn't do the help function here, I'm going to call this CT. CT for cut tree, and I'm going to cut the tree. And I'm going to eliminate one of my T's there. HC, and I'm going to cut it so that there are 5 groups. Okay, so it will do that for you. You could also specify a distance based on what you were visualizing there, but I've got my KT here. And now I have my different groups here. If I cut it so that I want 5 groups, it's going to give me 5 groups back. Okay, and if I want to look at a silhouette plot for that, I can do that, of course, by putting in now this information, CT. These are my groups, and my distance matrix, which is D. Okay, so this is what it's going to do. So this is flexible. It will work with any clustering method. You don't need to know the labels. You're just looking at that intercluster dissimilarity. So now what I might want to do is quartz plot SI. Okay, so this looks pretty bad, right? I mean, here I have negative numbers here and so forth. So this is looking like a really bad clustering. And of course, we know that 5 clusters is too high for this data set. But if you didn't know that, you might come up with something like this. So now let's do the same thing again. I'm just going to copy and paste it down here. And let's just take a look at how this thing hangs. And so now let's cut it at 1.95. Okay. So that one I'm going to use my height, 1.95. I'm going to do the same thing, maybe for comparison person pieces here. I want to have this, change these. It's good practice not to write over things like that. Okay, so now I have 3 groups. This looks a lot healthier. I still see some things that don't quite fit, okay? My silhouette plot is a 0.52. And I bet if I had 2 groups, it would probably look the best. So let's just take one more look. But this is in practice kind of what you can do. Obviously, it's a little bit easier to look at this than it is to say I want 5 groups. Okay, so to look at this and look for a height. So if I want 2 groups, I can take any value in this range will give me 2 groups, right? Anything. So let's just take 3. Okay, so that's my third cut, my third silhouette. Yeah, so this looks even better. Okay, so we see our 2 groups. They're a little bit uneven, but we have an average silhouette of 0.66. Okay, so the silhouette plots are telling me here, even with the hierarchical clustering, I could use it in connection with k-means, which I didn't show you. K-medoids I did show you. We can use it here with hierarchical clustering. It's independent of the method you're using. It's just going to give you that information. It's telling us there's 2 clusters in the data set. Okay. So now what I think I want to do is I want to show you Oh, maybe this is excessive or maybe it's not. Let's just do one more quick thing before we move on. And that is to write a little for loop, okay? If you want to take a look, for example, at how we might cut the tree over a series of values, okay? Maybe k is equal to 2, 3, 4, 5, 6, okay? So the first thing I'm going to do is I'm going to create a vector. I'm going to call it store, and it's going to be empty. And then I'm going to go for I in 2 to 6. Okay, so I'm going to march through with k is equal to 2, 3, 4, 5, and 6. And all I'm going to do is generalize this. Okay, so let's copy and paste this in here. I'm going to call these the originals now. KT, and I'm going to cut it not at height, but I'm going to cut it according to the groups. So K is equal to I in this case. So I is going to be 2, and then it's going to be 3, and then it's going to be 4, and then I'm going to plot my silhouette accordingly. And then what am I going to store? Well, I'm going to store the average width, okay, of the silhouettes, which is going to be that summary value, right? That was my 0.66 or my 0.53. It's the average of those clustering dissimilarities for the different clusters. And so actually, if I do summary, SI3, for example, my last one that I did here, I'm going to get all the information here for the different measures that I have here. And in fact, if I look at I should be able to pluck it out here. SI3, let's see. Let me try it like this. Names. Okay, so what's coming out of the summary object? Okay, perfect, that's it. So what I'm going to be wanting for here is the average width measure. Okay, so if I do summary SI3, AVG width, now I have my information that I want to track. Okay, so that is coming out of the summary object. It has a bunch of fields and I want the average width one. Of course, this is SI because I have generalized it now. So this is what I have, and now I want to store the values here. So I have store, and then I have every time I go through the loop, I'm going to pin on the average width. Okay, so let's take a look here, see if this runs, and let's look at my objects. Okay, so this is really telling me the silhouette, average silhouette plot. We can see it's going to start to get worse as we move up in K. The best one in this case is going to be K is equal to 2. Okay, all right. Okay, so the last example I want to share with you today is a little bit more complicated. Here we want to actually cluster high dimensional data. Okay, and we don't want to necessarily do that random sampling thing just so we can visualize things. So we probably should come up with a better way to do this. Okay, so what I want to do here is I want to look at something called the, it's a gene expression data set, and it is in the package, I can't remember what package it is, you have already loaded it, but it's called Golub MALT-test. Okay, so this is the data. It has roughly over 3,000 genes and 38 tumor samples. And so what we want to do is do some clustering of these samples, these 38 tumor samples, and we want to cluster them using this gene expression data. Okay, now there's some interesting fields here. If we just kind of look at the data, Golub I don't even want to do head in this case. I just want to look at the first 5. Oops. Sorry, I have to bring it into the field first. Data, GoLoop. Okay, now I have loaded it into the field. And actually, I want to bring this over here so I don't forget. And so what I have here is, if I look at GoLoop, the first 5, this is the information, dim, GoLoop. God, I cannot spell today. This is the rows are the genes and the columns are the sample labels. So C1, this is going to tell me, actually this is telling me the status, okay? And code 0 is ALL, which is a certain type of leukemia, and the other codes are for another type of leukemia called AML. So these are the two. It's not healthy and disease, it's just 2 different types of leukemia. These are their labels. We can see there's many more of the acute, there's far fewer acute cases. Then we have the gene names, and the gene names, it's a big vector, and they're not actually gene names, they're just entree or ensemble IDs that can map to a name, so I won't show you those right now. So what we want to do here is we're going to do a t-test, and the way I'm going to do this is I'm going to write a function, and I'm going to call it my t-test. Okay? And it is a function, and it is going to consist of a t-test for x, okay, because this is a function, so I'm writing it in general, and it's going to be 1 to 27, the first 27, so x is going to be a vector in this case, okay, and x going from 28 to 38. So you can already figure out what I'm doing here probably. I'm doing a t-test according to the 2 different types of leukemia here, which are contained in the first 27 columns and the last 11 columns. So this is what I want to do now. I want to apply this to the rows of my data. So in other words, I want to march through all 3,000 genes, and I'm going to do a t-test for each one of these rows to determine if there are significant differences in the mean of the expression profile for each gene between these 2 cancer types. So it sounds like a mouthful. There's a reason why we're doing this, okay? And so actually, if you look at t-test, okay, most of you know how to do the t-test. The output here, if we come down to value, we can actually grab the p-value from this. I don't want to output the entire t-test object. All I want to do is stack up the p-values. So I'm going to do this. I'm going to put a $sign and okay, so it's going to do the t-test. I'm going to put in this general x. You can see, so instead of writing a function, I could have done a for loop where I crawl through all 3,000 in a big for loop. This is going to be a more efficient way to do it, although it's still, with this number of genes, it's not super taxing anyways. But I'm going to create a field called p-value, and I'm going to apply one of the most handy functions in R. I'm going to apply to my GoLoop data, which I've loaded, 1, which means I'm going to use the rows. To every single row, I'm going to apply a function, the function that I just wrote, my t-test. Okay, so that's what I have. And now I have sorted p. I'm going to order my p-values. Okay. Okay, so let's just take a look. Let's see what this looks like. Okay. Oops. P-val. Okay, so sorted p Okay, so these are ranked in order. This means this is the smallest, this is the next, and so forth. And what I want to do here is I just want to grab the most significant ones. So this is one way to kind of sort things out. So golub.order. I'm going to simply take my original matrix golub and I'm going to shuffle it around such that the most significant things are sitting at the top. So I'm going to create this thing and it's going to be called golub order and it's going to be stacked according to the most important ones being on the top. Okay, so this is convenient. I'm reshaping the matrix accordingly. And if I look, okay, so if I look at dim golub.order, it's still the same size. Okay, I'm just reshuffling. I've done my t-tests and I know what the most significant ones are. Those at the top of my matrix now are the genes that are changing the most. Okay, they have the smallest p-value. So let's just grab a few things. I'm going to call it DE genes. This is going to be golub.order. I'm just going to take the top 250. Oops. Okay. So I'm taking the 250 most significant genes. Okay. And maybe what I want to do first of all is write a table of DE genes. Okay, I'm just going to call it that, and I'm going to use a tab delineated file, and the row names are going to be row names DE genes. Okay, and the col names, and I need a dot here, I always forget, are going to be the col names of DE genes. Okay, so here we have this information, and the only thing I forgot to do up here is I forgot to name. Okay, so I want to create, I want to kind of fancy up this golub thing. So I want to create a row names for this data set, and I want to do that. I want to call it the actual gene names. So golub.gnames, and it's actually the third. Okay, so if I look at this, these are all the gene names. That's like what I said. They're not actually gene names. This underscore AT is specific to the platform that this was measured on, but these are all accession numbers, so you can actually use these and do a batch query to look at how these things map. And I'm not sure, actually. Yeah, so these are all the AFI things. Okay. So this is what we have, and that's going to be my row names and my col names. I'm just going to call this paste. Excuse me, my text messages are showing. So C, I'm going to paste ALL. That's the name of the first type of leukemia, 1 through 27. And I'm going to separate that with a So this is what I'm going to do for the first bunch, and then the next bunch, I'm going to make this one a ML, and I'm going to call this 28 because it is the 28th sample that I have, and I'm going to send this to 38 to correspond to the columns. So now what I have done is the same thing. I have just created this. If we wanted to, we could run it again. And now hopefully when I write my file, I have a, forgot my underscore here. Actually, what I'll do is this. Now if I open this, okay, so if you open your data file, you'll see a smaller subset of these data. You can look right quickly here at DE genes 1 through 5, 1 through 5. And you can see they contain the same labels that the GoLoop dataset will contain as well. Okay, so let's get down to the business of clustering these things. I'm going to use a function called heat map. There are two heat map functions. One is called heat map. The other is called heat map 2. Okay, there's just two versions. Let's just open both of them actually. Okay, so heat map 2 actually must be in ggplots. Okay, so if you I'll eliminate this for now because we're not going to use ggplots. But if you look at the ggplots version, you'll see it's almost the same thing, just with some different options for the visualization. There's a lot of things that go into this. The basic things that you need to know is you need to specify the colors that you want to use and label things. So let's do heat map DE genes on my smaller set. And I'm going to use colors. I'm going to use the CM colors. There's lots of colors here. I'm going to use a range of 20. And my X label is going to be conditions. And my Y label, I could also say samples, is going to be genes. Okay. This is going to take a minute to plot. Oh, not as small as I thought. Okay. So the genes are plotted on this axis. You can see it's a very crowded plot. Obviously, the visualization is not really feasible. This is for 250, but I can see the conditions or the samples, whatever you want to call them, every time I kind of, I guess we can't see very well in this case, but I can tell just by the font here that these AML seem to be grouping together. We can see a much more prominent pattern in these samples. Look at how this looks. There's this bunch of genes here, if whatever these are, that are really looking quite distinct from the other ones. And this is really, it's this block actually that's helping discern, if I look at this dendrogram, this group of samples to this one. It's really, we can tell in this plot, which is why I did this analysis, that it's really these things. Now, if you were to take a random 250 genes from this data set and cluster them, you wouldn't see a dendrogram like this. Okay, but what we have done is we've done some pre-selection. We did pre-selection based on a t-test. Okay, that's one way. So those things that are coming up at the top, which we've sorted and truncated our matrix accordingly, these are the things that are changing the most because these are the ones that are separating the two means. This is the significant difference between the two means between these two groups. So if you ever stand a good chance at seeing separation between groups in a data, it's going to be either through a low variance filter or by filtering on a t-test. Okay, so here we see really beautiful separation. Okay, so that's all I wanted to show you for today. There's lots more in your textbook, the Introduction to Statistical Learning on clustering and so forth, but I think this should be a pretty comprehensive start.